Hello, very good evening to the students who have joined. Can you let me know in the chat box if my voice is audible and if my video is visible to you? So, okay, thank you. Please let me know if the presentation is visible. The presentation slide is visible to you? Okay, thank you so much. Now we will wait till more students join and then we will start today's session. Okay, so it is 6 p.m. I will wait for 2 more minutes and then I think we can start the session. Eventually, when more people will join, I will add them up. Okay, so I think I will start with the introductions. So, good evening to the students who have joined. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am the TA for this NPTEL course of Organic Chemistry in Biology and Drug Development. Today it is our week 8 live session, and in this week 8 live session I will discuss the course content which is in the week 9 for this particular course. So we are going ahead with every week. So today is week 8 live session where we will discuss the week 9 course content. So before starting the session, I would like to clarify few points, but I think not much students have joined. So let us wait for 1 to 2 more minutes, then we will start the session. Let us wait for 2 more minutes and then we will start the session. Okay, so I think the presentation is visible and my voice is audible. If you have any problem, please let me know in the chat box. If there is any problem regarding my voice or the presentation, please let me know in the chat box. So again, I'm Mousumi Deb, the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad, and today it is the week 8 live session for the NPTEL course of Organic Chemistry in Biology and Drug Development. So in this class, what we will do is, so before starting the class, I will like to remind you a few points. So there will be a problem solving sessions every week. Okay, for this NPTEL course of Organic Chemistry and in Biology and Drug Development, for 12 weeks it will be. These sessions will be on every Tuesday from 6 p.m. to 8 p.m. And the sessions, these will be live on Google Meet platform. If any network disruption happens during the class, please wait for a few minutes. Please do not leave the session. I will try to join as soon as possible. If the internet disruption happens at my side, I will try my best to join as fast as I can. And if the network disruption happens at your side, you don't need to be worried about it, because I will share this lecture video and also this PPT with you. Okay, so you can check it after the session also. So during the session, the students can post their question and doubts and query through the chat box. Okay, and in between the questions, in between the session, I will look at the chat box every time and I will answer the doubts and queries of the students. For smooth streaming of the class, I have just muted the mics and also the camera for this of the students, so that the class there is a smooth streaming of the class. So if you have any doubt or if you want to share anything with me, you can drop that query in the chat box. And if still any doubt persists after class, the students can drop an email to me. I will share my email ID at the end of the session, and you can actually drop an email to me regarding any point or any topic after the class. Okay, I will answer the query through email. And this presentation and class recording, as I have said before, it will be shared to you after the session. Okay. So today it is the week 8 live session for this course, and in this week 8 live session, what I will do is, I will discuss the week 9 course content. Okay, so right now the week 9 is live, so I will discuss the week 9 course content. We will solve today around 20 MCQs regarding the week 9 course content. Okay, and this 20 MCQs, first I will show you the question. You have to try to answer the question of that particular question, and there are 4 options for each question, and you have to write whatever you think is correct in the chat box. Okay, I will check after I show you some question. I will give you some time, and then I will check what is the answer is given by you, and then what I will do is I will show you the correct answer, and then we will discuss the questions. Okay, so what is the week 9 course contents? Let us discuss that first. So in this week 8 session we are solving the week 9 session questions, because week 9 is live right now. So what are the topics that are included in the week 9? The topics are: number 1, introduction to drug discovery process. Then we will discuss fundamental principles of drug development process. Then combinatorial chemistry. Then neurotransmitters. Then catecholamine based and GABA neurotransmitters. Then hypertension, humoral mechanism and renin-angiotensin system. And inhibitor design of angiotensin converting enzyme, or ACE. So these are the topics which are involved in your week 9 course content, and these topics are very important for your drug development and your the role of organic chemistry in drug development, which is your course is NPTEL. So this starts basically from here, because these topics like drug discovery process, fundamental principles, these are now we will start about it. So let us start the session. Okay, eventually when more students will join, I will just let them in. So let us start with the first question of today's session. So now what is the first question of today's session? So problem number 1, it says that which of the following statements is true? Your options are: number A, drugs and drug targets have similar molecular weight. Number B, molecular weight of drug target is higher than that of the drug. Number C, molecular weight of the drug is higher than that of the drug target. And number D, there is no general rule regarding this. So we are discussing the drug development process today, and the very first question is regarding the drugs and drug targets. So read the all options carefully and let me know in the chat box which of the following option you think is true regarding the drugs and drug targets. Your options are: number A, drugs and drug targets have similar molecular weight. Number B, molecular weight of a drug target is higher than that of the drug. Number C, molecular weight of the drug is higher than that of the drug target. And number D, there is no general rule regarding this. Okay, so I have one option answer which says option number C. I will give you 2 more minutes. Everybody, I want all of you to try, because there is no marks regarding these questions, but if you try to answer, it will help you. Okay. So which of the following statements is true? Read all the options very carefully, then answer it. Okay. Your options are: number A, drugs and drug targets have similar molecular weight. Number B, molecular weight of the drug target is higher than that of the drug. Number C, molecular weight of the drug is higher than that of the drug target. And number D, there is no general rule regarding this. Take some time and then I will discuss it. Everybody try to answer this question in the chat box. Which of the following options you think is correct? Okay, so I have till now 2 answers, number C and number B. So now let me check which of the following is correct answer. So the correct answer for problem number 1 is number B. Molecular weight of a drug target is higher than that of the drug. Now let us discuss a drug. What is a drug? Now a drug is a chemical substance, natural or human made, that changes normal body functions in some way. There is no set of definition for the term drug, but it is one we use in the class. So what is the drug? Drug is a substance that is natural or human made, and that changes the normal body functions in some way. And there is no actually particular definition regarding the drug, but drugs are used to cure or treat the disease, reduce symptoms of illness, alter a person's mental or physical state. So the chemical substance, the particular chemical or the particular molecule which alters the normal body functions in some way, it may be for a cure, or treat a disease, okay, or reduce the symptoms of a illness, or alter a person's mental or physical states. That substance, that chemical or that molecule can be termed as drug. Now what is the drug target? So basically drug targets are the various human body mechanisms that are targeted by this particular drug. So these may be proteins, okay, so these may be enzymes, DNAs, miscellaneous biomolecules, other receptors, okay, ion channels, transporters, nuclear hormone receptors. So these receptors, this protein, ion channels, or the bodily targets where a particular drug can bind, or it can act or it can exert its activity, it can be termed as a drug target. So your options were number B was a correct, which says the molecular weight of drug target is higher than that of the drug, which is true, because the drug targets mostly are complex protein molecules or DNA or integrins or miscellaneous biomolecules, receptors, ion channels. So already these all biomolecules have a high molecular weight very high, okay, but the drug, which is a very small chemical substance or molecule, it will have the particular molecular weight which is very low, okay, than that of the drug target. So the correct option was number B, which says that molecular weight of a drug target is higher than that of the drug, okay. And the drug targets are like enzymes. Enzymes consist of 47% of normal drug targets. We have DNA, which consists of 1%, integrin 1%, other receptors 4%, GPCRs 30%, ion channels 7%, okay, and transporters 4%, nuclear hormone receptor 4%, and miscellaneous 2%. So these are various your drug targets. So remember, molecular weight of drug targets is higher than that of the drug. So number B is correct, okay. So now I am moving toward the next problem. Again, if you have any doubt, any query, please let me know in the chat box, okay. So problem number 2 says, what is meant by a lead compound in medicinal chemistry? Your options are number A drug containing the element lead. Number B, a leading drug in a particular area of medicine. Number C, a compound that acts as a starting point of drug design and development. Number D, a drug which is first to be prescribed to a particular for a particular ailment. Again, I am repeating, what is meant by a lead compound in medicinal chemistry? Your options are number A drug containing the element lead. Number B, a leading drug in a particular area of medicine. Number C, a compound that acts as a starting point for drug design and development. And number D, a drug which is first to be prescribed for a particular ailment. Now, what is meant by a lead compound in medicinal chemistry? Read all the options carefully and try to answer this in the chat box. I want all of you present in the class try to answer this question. Number A, B, C or D, which of the following is correct? There is no marks given for this kind of these questions, but so you can just doesn't matter if your answer is wrong, but all of you please try. In that way you will understand these topics more better. What is meant by a lead compound in medicinal chemistry? So, what is the answers? Number C. I have one answer, number C. Okay, others also try. Okay. I will give you 2 more minutes. Which of the following is meant by lead compound in medicinal chemistry? A drug containing the element lead, a leading drug in a particular area of medicine, a compound that acts as a starting point for drug design and development, and a drug which is first to be prescribed for a particular ailment. Which of the following is meant by the lead compound in medicinal chemistry? Yeah, I think there is only one answer. So let me check again. Okay, number C, number B. Okay, so I have number C and number B most as an answer. Now let us discuss and let us see which of the following is correct. So the correct answer for this question is number C, a compound that acts as a starting point for a drug design and development. It is meant by a lead compound in medicinal chemistry. Now let us discuss. So what is a lead compound? A compound demonstrating a property likely to be therapeutically useful. The level of activity and target selectivity are not crucial. Used as a starting point for drug design and development. Found by the design, molecular modeling or NMR, or by screening compound, natural or synthetic. Need to identify a suitable test in order to find a lead compound. What is the active principle for lead selection? A compound that is isolated from a natural extract and which is principally responsible for the extract's pharmacological activity, often used as a lead compound. Okay, so what is a lead compound? A lead compound is a compound which acts as a starting point for a drug design and development. Okay, it can be a natural compound, okay, or it can be a synthetic compound. The synthetic compound can be identified by designing like molecular modeling or NMR, or by screening compounds which can be natural or synthetic. And what is the principle is that this compound must be from a natural extract and principally responsible for the extract pharmacological activity. So what is the start of this drug discovery cycle? So basically first the compound collections, okay, and primary assay, the high throughput and in vitro assays can be done. Then we have secondary assays like counter screens, bioavailability, toxicity, metabolism. These give us a lead compound and we have to perform SAR on this lead compound. Then we have to perform the structural characterization of protein ligand complex, which can be direct or indirect lead to the design. Then chemical synthesis. Then again the primary assays, secondary assays are to be done, and after this cycles we have this clinical candidate. So we will discuss this thing in more detail later. So don't you have to worry about it. So remember, what is a lead compound? It is a compound that acts as a starting point for a drug design and development. Okay. So now I will move toward the next problem. Problem number 3 says, in drug discovery process, pharmacokinetic study is done during. Your options are number A, basic research, number B, post market surveillance, number C, preclinical study, and number D, clinical development. So in drug development process, pharmacokinetic study is done during which period? During the basic research time, or during the post market surveillance, or during the preclinical study, or during the clinical development? Try to answer this question in the chat box. Then I will discuss what is the drug development process, and then the pharmacokinetic study, what is the pharmacokinetics, I will discuss in detail. But before that, I want all of you to try to answer this question. When pharmacokinetic study is done during a drug development process? Everybody present in the class, try to answer this question. Which of the following option A, B, C or D you think is correct? Write it in the chat box. So, when pharmacokinetic study is done during the drug development process? Let me check the chat box. So, I have few answers like number D, number A, number D and number C. Okay, so I have, I think, different options here. So, let me check. So, the correct answer for this question will be number C, preclinical study. So, during preclinical study, the pharmacokinetic study is done. So, what is drug development process? So, this is the cycle for the drug development process, and it is a very long cycle. Okay, drug discovery and development process. So, first you have to select a disease target. The disease you want to, there are several diseases. Okay, so first you have to select the disease and then you have to select the disease target. So, what is the difference between a disease and disease target is, so there is every disease like let us say cancer. So, these kind of diseases have several way by which it can be tackled. Okay. So, there are several various paths by which a particular disease can be treated, and to select a particular path or to select or we can say like there are various drug targets for a particular disease. Okay. So, a particular disease can be treated by targeting a particular various drug targets. So, selecting a particular drug target is the first thing before starting a drug development process. Okay. Like for example, cancer, it can be treated by targeting a particular enzymes which are very important for the survival of the malignant cells. Okay. So, this is a one approach. So, it can be treated, the cancer can be also targeted by treating the or by inhibiting by DNA synthesis of the particular malignant cells. So, DNA can be one target, different enzymes can be one target. So, first we have to select the disease target, the disease which we want to treat, and the target for that particular disease which we will use for our discovery of the drug. Okay. Then first we have to do is the basic research. So, what is the basic research? What are the things? We have to do the target identification, target validation, assay development, genomics and proteomics. Okay. Then we have this early discovery, and what happens in this during the early discovery? We have computational studies, high throughput screening, identification of hit to lead and lead optimization. So, all of this happens during the early discovery. Now, we have preclinical studies. So, what happens during this preclinical studies? We have in vitro studies, in vivo studies, pharmacokinetics, formulation, dose and dosage studies. Then after this all basic research, early discovery and preclinical study, then we will go toward this clinical development phase. And after clinical development, we have this FDA approval. FDA is the Food and Drug Administration, and it is a USA based organization which actually approve the drugs. If you buy a medicine, you will see at the back side it is written that FDA approved. Okay. So, and then we have this post market surveillance. So, what happens during post market surveillance is you have launched a new drug in the market. So, after 1 year or 2 year, we have to monitor what is the side effects, if any, for the drug on a larger community of the people. Okay, and that is called the post market surveillance. If the drug is found to be safe for the after the post market surveillance, it is kept in the market only, and then otherwise if it is found to be harmful for a larger community, then it is withdrawn. Okay. So, the preclinical study, during the preclinical study we have this pharmacokinetics. Now, what is meant by this pharmacokinetics? So, pharmacokinetics basically we can say like what our body is doing to a drug. Okay. So, what is the pharmacokinetics? It is called the ADME. So, what is ADME? So, we have ADME. Okay. So, what is ADME? A for absorption, okay, D for distribution, M for metabolism, and E for excretion. So, these are the 4 steps for the pharmacokinetics. Okay. So, first a drug is administered, okay, dose of drug is administered. So, it is input in the system. So, drug concentration in systematic circulation. So, it is then is the absorption. So, first you are taking a drug. So, first what we have it will have. So, our body what it will do, it will absorb the particular drug, right. Then we will have the distribution. So, this particular absorbed drug will get distributed in different tissues and blood circulation and different area of targets in our body. And then we have metabolism. The drug particularly will be metabolized by various organs, and elimination. The drug is after the metabolization, the excess parts of the drug which are not necessary for our body, it will be excreted. So, all these 4 portion, the absorption, distribution, metabolism and excretion is known as pharmacokinetics. So, pharmacokinetics is basically what? It is the what our body is doing to a drug. Okay. And again we have another term which is called the pharmacodynamics. So, what is pharmacodynamics? Pharmacodynamics is the like what is the drug is doing to our body. Okay. So, various pharmacological effect, clinical response like toxicity or effectiveness, these are part of a pharmacodynamics. Okay. So, we will discuss pharmacokinetics and pharmacodynamics in detail later in today's session or upcoming weeks also. So, just remember pharmacokinetics, it is the ADME, absorption, distribution, metabolism and excretion is the steps which our body is doing for a particular drug which is taken, and pharmacodynamics is the effects which is shown by the particular drug. Okay. So, this was the problem number 3. So, now I will move toward the next problem. So, problem number 4, it says that when a drug is approved for human testing during clinical trials. Your options are number A, between phase 3 and phase 4, option B, between phase 1 and phase 2, option C, at the end of phase 4, and option D, at the end of preclinical trials. So, when a drug is approved for human testing during the clinical trials. Try to answer it in the chat box. Which of the following options you think is correct? Option number A, B, C or D. Then I will show you the answer and we will discuss again. So, what I showed you in the last slide was the half of the drug discovery process basically, and half is remaining till now, which is known as the clinical developments, which is a very lengthy process again. So, try to answer this question in the chat box. Which of the following options you think is correct? I will give you 2 minutes. So, is there any answer in the chat box? think I have few answers, number C, number A, number B. Okay, now let me check which of the following is correct. So the correct answer is number D, at the end of preclinical trials. Okay, so before discussing it, let me show you the last one. So this is the half of the drug development process. Okay, so first is the disease target, then we have basic research, early development, preclinical study, and then clinical development starts. Now what is the clinical development phases? So this is the clinical development phases. So we ended there with the preclinical studies, right? So what happens during the preclinical study is the exploring if and how the new drug may work. Okay, it is done up to the preclinical studies. Then we have this phase 1, and we have 3 phases for this drug development before FDA review. So what is the phase 1? Phase 1 is happening like exploring the safety and dosing of the drug, and between the at the end of the preclinical study, between phase preclinical and phase 1, we have this drug approved for human testing. So if a drug is found to be safe enough after the preclinical studies, where this drug is approved for the human testing. Okay, then the drug will pass the 3 phases of human testing. It is the phase 1 when the drug is explored for the safety and dosing. Okay, the phase 2, the exploring the effectiveness of the drug. Phase 3, exploring the safety and effectiveness compared to currently available treatment. Okay, and then we have this FDA review. So what happens during FDA review? To confirm the safety and effectiveness of the drug. Okay, so at the end of the phase 3 clinical trial, the drug is submitted to the FDA for approval, and if the drug is approved after the FDA review, we have this drug in the market. Okay, then we have a phase 4 of clinical trial, okay, which is also called the post market surveillance, which I discussed this in the last slide. It is evaluating the drug over time and in a large number of patients. So all these preclinical studies to the phase 4, it takes 5 to 15 years. Okay, so the drug development process, it is basically very much time consuming. It takes at most of 5 to 15 years for a new drug to come into the market. Okay, and a special designation can speed up the process, means if there is a special necessity, like we have seen during the COVID times, it was a very the drugs were very fast, it was screened, and when the drugs came to the market very fast. So this was a special designation, right? It was a special need, but average it takes 5 to 15 years for a drug to come into the market. So remember, a drug is approved for human testing during the at the end of the preclinical studies. Okay, and remember this also, the drug is approved submitted to FDA after the phase 3 clinical trials. Okay, and FDA review happens in between the phase 3 and phase 4. Okay, so remember what are the phases, what happens during each and every phases, and also what happens in between these phases of clinical trials. So now next question. Okay, problem number 5 says, which of the following statements is true regarding Lipinski's rule? Your options are number A, log P less than 5, number B, molecular weight greater than 500 Dalton, number C, hydrogen bond donor greater than 10, and number D, hydrogen bond acceptor equal to 20. So which of the following statements is true regarding Lipinski's rule? Lipinski's rules is basically is a rule for drug likeliness, means a compound which will show more the drug like properties, and it can be defined by Lipinski's rules. So there are several rules. So you have to identify which of the following statements is true regarding the Lipinski's rule. Your options are number A, log P less than 5, number B, molecular weight greater than 500 Dalton, number C, hydrogen bond donor greater than 10, and number D, hydrogen bond acceptor equal to 20. Try to answer this question in the chat box. Which of the following you think is true? Then we will discuss what is Lipinski's rule and what are all these terms like log P, what is molecular weight and everything. Try to answer this question in the chat box. All of you should try to answer these questions. Even if your answer is wrong, but trying is important, otherwise I won't understand if you are understanding the topics or not. Which of the following statements is true regarding Lipinski's rule? Let me check the chat box now. Okay, so number A, number C, number C. So I have 2 number A and I have 2 number C. Okay, let me check which of the following is correct and then let us discuss. So the correct answer is number A, log P less than 5. Now let us discuss. So Lipinski's rule, also known as Lipinski's rule of 5. Okay, so it gives us about the drug likeliness of a particular compound. Okay, so what is the Lipinski's rule? Not more than 5 hydrogen bond donors. So hydrogen bond donors should not be more than 5. Okay. Not more than 10 hydrogen bond acceptor. So number of hydrogen bond acceptor should not exceed 10. Molecular weight less than 500 Dalton. Okay, so molecular weight of the compound should be 500 Dalton, and C log P or log P value should not be greater than 5. So this C log P or log P known as the lipophilicity of the particular drug. So this particular value should not exceed the value 5. So this one is correct. Okay, log P less than 5, it is correct. Molecular weight greater than 500 Dalton, it is not, because molecular weight should be less than 500 Dalton. Hydrogen bond donor should not exceed 5. Okay, and hydrogen bond acceptor should not exceed number 10. So this Lipinski's rule, also known as the compound's drug likeliness. So if a compound or particular molecule follows all these four particular rules, it is known to be a good candidate for a drug. Okay, so what is P here? So let us discuss now what is log P or what is P. Okay, so P is the partition function. So it is the measurement of the drug's water solubility. Partition means the drug will be divided into in parts between water and oil layer. Okay, so P is the concentration of a compound in O, or O means octanol. Okay, octanol means it is an organic layer, and divided by concentration of drug in a water layer. Okay, and log P is the log concentration of compound by this. So this is the particular value. Okay. So basically what happens, so when you are this is called the separating funnel. This instrument is called the separating funnel. So if a compound is taken in this funnel, and we have added water and organic solvent. Okay, organic solvent may be any organic solvent there may be. So if and we shake this compound, so what happens, this compound distribute itself in between this water and this organic layer. Okay, so based on the density of the particular organic compound, it can be on the top of this separating funnel or in the bottom. So like if you have a organic solvent like DCM, okay, or dichloromethane, so these compounds are more heavier than water. So this density of DCM is heavier than water. So if we take the DCM and or DCM as organic solvent, it will be in the lower portion, okay, and water will be on the upper portion. But if the organic solvent is have less density, like ethyl acetate, it has less density than water. So ethyl acetate will be on the top and water will be in between in the lower portion. Okay, so based on the density of a particular solvent, okay, particular organic solvent, it can be on the top of the separating funnel or it can be in the bottom of the separating funnel. Okay, so a compound, particular compound, it actually when you have taken the compound in the solution, it distributes itself between the water and this oil layer or organic layer. Okay, so we have this formula for partition coefficient. So what is the formula for partition coefficient? It is the P or this log P. So log P, we have this formula equal to log of, so what is P? P means concentration of the compound in organic layer or oil layer and or hydrophobic layer, and in the concentration of compound in the water or hydrophilic layer. Okay. So, if the log P value greater than 2, okay, then the drug is lipophilic drug. Lipophilic means it has more affinity toward the hydrophobicity. Okay, it is more hydrophobic or it has more affinity toward the hydrophobic layer. And if the value of log P is less than 2, it is the hydrophilic layer. Hydrophilic means, hydrophilic drug means it is more water soluble. Log P only applied for neutral compounds. Low log P means low penetration to CNS, CNS means the central nervous system. High log P means low water solubility. It is the drugs are not suitable for oral administration if it has a very high log P, because it will not be soluble in water so much. So, we have to give this kind of drugs intravenously. Okay. So, there are few exceptions for this Lipinski's rule. This is called the plexitaxel. So, paclitaxel is a anti-cancer drug. It is a natural product. So, various natural products which are acts as a drug, so they do not follow this particular Lipinski's rule of 5, because natural products, they have very high molecular weight which exceeds this 500 Dalton rule. So, or this other rules also. So, remember this is very important. Okay, remember this thing, remember the particular exceptions for the Lipinski's rule. Okay, the drugs, this plexitaxel, vincristine, evermectin, so these are the various natural product or sugar based drug. So, these do not follow this Lipinski's rule of 5. Okay. So, now I will move toward the next problem. Problem number 6 says, identify the correct statement about the combinatorial chemistry. Your options are: number A, one molecule can be done in one time. Number B, it has high risk of failure. Number C, it was developed using solution phase synthesis. And number D, it creates an impact in lead discovery and optimization. So, identify the correct statement about the combinatorial chemistry. Read all the options carefully and write in the chat box which of the following you think is correct. Even if your answer is wrong, it does not matter, but all of you should try to answer this question. After your trial, I will just show you what is combinatorial chemistry and we will discuss it in detail. Okay. So, what is correct statement regarding combinatorial chemistry? Number A, one molecule can be done in one time. Number B, it has a high risk of failure. Number C, it was developed using solution phase synthesis. Number D, it creates an impact in lead discovery and optimization. I will give you few more minutes, then I will discuss it. Everybody present in the class, please try to answer these questions. Even if your answer is wrong, it does not matter, but all of you should try to answer. Okay. Okay, let me check the chat box now. Okay, so number D is most given answer. Okay, let us discuss. So the correct answer is very good. Number D, it creates an impact in lead discovery and optimization. So what is combinatorial chemistry? So combinatorial chemistry is a technique by which large number of different but structurally similar molecules are produced rapidly and submitted for pharmacological assay. This technique uses the same reaction condition with the same reaction vessels to produce a large range of analogues. The technique invented in late 1980s and early 1990s to enable tasks to be applied to many molecules simultaneously. Combinatorial library, the products produced from a combinatorial chemosynthesis are known as combinatorial library. These libraries may be a collection of individual compounds or mixtures of compound. Screening the components of a library for activity using high throughput screening techniques enables the development team to select suitable compounds. Okay, so what is combinatorial chemistry? It is a technique by which large number of different and structurally similar molecules are produced rapidly and submitted for pharmacological assay. So combinatorial chemistry within drug design. So what is there is various aspects of drug design where combinatorial chemistry can be applied. Okay. So these are therapeutic target to lead discovery, to lead optimization, to development candidate, to drug. So these are the various steps for a drug design, and this combinatorial chemistry can impact during the lead discovery and lead optimization. Okay. And what is the difference between the conventional drug discovery and combinatorial drug discovery? In conventional one molecule at a time can be screened, but in combinatorial many molecules at a time can be screened. So it is the make, purity and test for the conventional, but the combinatorial make, test and then purify. Hundreds of molecules can be done by the conventional method, and thousands of molecules in a month can be done by the combinatorial method. Slower lead generation in the conventional method, faster lead generation in the combinatorial method. High risk of failure happens during conventional method, but in the combinatorial the low risk of method, and both synergistically if it is used, the lead identification can happen more faster. So this is all about the combinatorial chemistry. Now I will move toward the next problem. Problem number 7 says solid phase synthesis is frequently used in combinatorial chemistry. What is meant by solid phase synthesis? Your options are: number A, reactions are carried out without solvent. Number B, reagents and reactants are attached on a solid phase support. Number C, reagents are used in solid phase. Number D, molecules are constructed on a solid phase support. So what is meant by solid phase synthesis? I think we discussed regarding the solid phase synthesis in our very first weeks, okay, where there was solid phase peptide synthesis. So the solid phase synthesis is very much frequently used during the combinatorial chemistry. Now identify which of the following statements more appropriately describes the solid phase synthesis. What is meant by solid phase synthesis? Let me check the chat box. Okay, so I have B, number D, number D. So I have 1 number B and 2 number D. Okay, let us now discuss. So the correct answer is number D. Molecules are constructed on a solid phase support. Now what is a solid phase synthesis? Let us discuss. So basically combinatorial synthesis is based on solid phase chemistry. It uses filtration as a separation and purification technique. The easy removal of unwanted substance by a simple filtration is the core of the library synthesis. Okay. So it is this synthesis substituted resin beds are used as a solid phase, which is actually a gel like matrix of connected polymeric molecules distended by the axis of the solvent molecules. Okay. So basically what happens, so there are resin beds, okay, and upon this resin bed, this is a polymeric kind of resin bed, and upon this resin bed which is used as a solid support, the molecules are constructed and filtration and used as a separation methods. Mean the molecule which is constructed, it is attached with the resin bed, okay, and if we wash the particular resin bed with the solvents, which will not detach the particular molecule from the resin bed, but it will actually remove the unwanted substance by the easy removal, okay, the easy filtration. So this combinatorial synthesis or combinatorial chemistry, this solid phase synthesis is used very much. So what is the resin beds? So the use of solid support for organic synthesis requires a cross-linked insoluble but solvent soluble polymeric material that is inert to the condition of the synthesis. So some means of linking the substance to the solid phase that permits selective cleavage of the product from the solid support during the synthesis, a synthetic procedure compatible with the linker and the solid. So these are the points which we need to focus during for choosing the resin beds for the solid phase synthesis. Okay. So the resin support should be cross-linked insoluble but solvent soluble polymeric material that is inert to the conditions of the synthesis. Some means of linking the substrate to the solid phase that permits selective cleavage of the product from the solid support during the synthesis. A synthetic procedure compatible with the linker and the solid. Okay. So this was all about the solid phase synthesis, which is frequently used in the combinatorial chemistry. Again I am saying, if you have any doubt, any query during the session, if you do not understand a particular question or particular answer, please let me know in the chat box. Okay, I will then and there I will let you know, or I will try to clarify your doubt. And even if you do not want to say it now, you can later at the end of the session you can tell me in the chat box, or you can drop an email to me. Okay, just note down if you do not understand a particular thing, and then at the end of the session let me know. Okay. So now moving toward the next question. Problem number 8 says, which of the following statements is incorrect regarding the blood-brain barrier? Your options are: number A, the walls of capillaries supplying blood to the brain have such have a such. So okay, this should not be have are such. There is a have such are such. So okay, the have won't be there. Okay, are such, which makes it difficult for polar drugs to leave the capillaries. So there is a mistake here. So it will be are, not have. So next option is, the capillaries in the brain have a fatty coating, making it more difficult for polar drugs to enter the brain. Number C, the inner walls of capillaries supplying blood to the brain are hydrophobic in nature. Number D, hydrophilic drugs pass through the blood-brain barrier more easily than the hydrophobic drugs. Now you have to identify the incorrect statement regarding the blood-brain barrier. Again I will repeat the options. Number A says, the walls of capillaries supplying blood to the brain are such which makes it difficult for polar drugs to leave the capillaries. Number B, the capillaries in the brain have a fatty coating, making it more difficult for polar drugs to enter the brain. Number C, the inner walls of capillaries supplying the blood to the brain are hydrophobic in nature. Number D, hydrophilic drugs pass through the blood-brain barrier more easily than the hydrophobic drugs. Identify the incorrect statement regarding blood-brain barrier. All of you read the options very carefully and then try to answer this question. After your trial, I will just show you the answer, then we will discuss what is the blood-brain barrier. Read the options carefully, then try to answer the question. Which of the following statement is incorrect regarding blood-brain barrier? Let me check the chat box. Okay, I have only one answer, number D. Okay, let us discuss about it. Okay, every student, I want all of you to try. Okay, even if your answer is wrong, it does not matter, but all of you should try to answer the questions. The correct option will be, very good, number D. Hydrophilic drugs pass through the blood-brain barrier more easily than hydrophobic drugs. It is incorrect. Okay, before going on, I will just try to correct this thing. Perfect. Okay. So, number D was the correct answer. Now, let us discuss what is blood-brain barrier. So, what is blood-brain barrier? The blood-brain barrier, or BBB, is formed by the single layer of endothelial cells that line the inner surfaces of capillaries in the brain. So, it is a semi-permeable capillary membrane, that is, it allows some materials to cross but prevents others from crossing. In most parts of the body, the capillaries are lined with endothelial cells. The endothelial tissue has small spaces between each individual cell, so substances can move readily between the inside and the outside of the vessel. However, in the brain, the endothelial cells fit tightly together and substances cannot pass out of the bloodstream. Some molecules such as glucose are transported out of the blood by the special methods such as active transport. So, what is the physiology in the brain? Endothelial cells are packed much tighter together due to the existence of zonula occludens, the tight junctions between them blocking the passage of most molecules. The blood-brain barrier blocks all molecules except those that can cross the cell membranes by the means of the lipid solubility such as oxygen, carbon monoxide, ethanol, steroid hormones. Those that are allowed by a specific transport system such as sugars and some amino acids. Substance with molecular weight greater than 500 Dalton generally cannot cross the blood-brain barrier. So, hydrophilic drugs generally cannot pass through the blood-brain barrier more easily. Rather, hydrophobic drugs, the compound which are hydrophobic or lipophilic in nature, they can pass the blood-brain barrier more easily. Okay. So, this was all about the blood-brain barrier. So, option number D, it is incorrect. Hydrophilic drugs cannot pass through the blood-brain barrier more easily than the hydrophobic drugs. So, remember what is the blood-brain barrier and what is the characteristic of this particular blood-brain barrier. So, it was the problem number 8. Now, I will move toward the next problem. Problem number 9 says SAR shows that important binding groups in morpholin are phenol, aromatic ring and amine. A diagram can be drawn which shows these functional groups and their relative orientation. What is the term for this? What is the term for this particular diagram? Okay. The options are number A, pharmacodynamics, number B, pharmacokinetics, number C, chromophore and number D, pharmacophore. So, what is the term which is used? Read the question very carefully. SAR shows that important binding groups of morpholin are phenol, aromatic ring and amine. So, a diagram can be done which shows these functional groups and their relative orientation. What is the term for this diagram? Your options are pharmacodynamics, pharmacokinetics, chromophore or pharmacophore. Now, read the question very carefully and then try to answer it in the chat box. So, is there any answer in the chat box? Okay, I have few answers, number B, number D. So, number D seems to be most frequently given. Let me check. Okay, so the correct answer is yes, number D, pharmacophore. So, pharmacophore is the term which is a diagram which can shows actually various functional groups and their relative orientations. Okay, so now let us discuss each and every term which is given as the options here. So, what is pharmacodynamics? Pharmacodynamics is the study of mechanism of action of drugs and explains what drugs do to a body and how do they do it. It tries to explain the action effect relation as well as dose relation effect. It involves the drug receptor interaction to elicit the action of drug in the body. Pharmacodynamics is the study of biochemical and physiological effect of drugs and their mechanism of action. It provides the basis of rational use of drugs and design of better effective drugs. So, pharmacokinetics, as I have also mentioned it before in the previous slides, like it is the what the drug is doing to our body, what are the pharmacological effects, what are the physiological or biochemical effects of the particular drug and what is the exact mechanical mechanism of action. Okay, it is known as pharmacodynamics. What is the pharmacokinetics? Pharmacokinetics is the quantitative study of the drug movement in, through and out the body. Okay, it is the study of the process by which the drug is absorbed, distributed, metabolized and eliminated by the body. The absorption, distribution, metabolism and excretion of the drug all involve is passage across the cell membrane. Pharmacokinetic properties are affected by the route of administration and dose of the administered drug. Okay, so what is pharmacokinetics? Pharmacokinetics is the quantitative study of the drug movement in the body. So, these two terms are very important, pharmacokinetics and pharmacodynamics. One is what is our body is doing to a drug and another is what is the drug is doing to our body. So, what is a chromophore? Chromophore is a Greek word. Chroma means color, phore means bearer, defined as any isolated covalently bonded group that shows a characteristic absorption of electromagnetic radiation in the UV and visible region. For example, the compound containing chromophore is called the chromogen. Like carbon-carbon double bond, carbon-oxygen double bond, nitro, these groups are known as chromophore, which actually increases a particular compound's absorption in the UV or visible region of the electromagnetic radiation. Now, what is pharmacophore? So, IUPAC definition is an ensemble of static and electronic features that is necessary to ensure the optimal supramolecular interactions with a specific biological target to trigger or block the biological response. In drug design, the term pharmacophore refers to a set of features that is common to a series of active molecules. Hydrogen bond donors, acceptor, positively negatively charged group and hydrophobic regions are typical features. We will refer such features as pharmacophoric groups. So, in a particular, basically in short, in a particular compound we have various functionalizations and these functional groups have several reasons, okay, or the several actions to toward the drug target. Like amine group, they can participate in hydrogen bond donation, okay, or carbonyl group can participate in hydrogen bond acceptance. So, these kind of functions they have with the drug target interactions. So, these are known as the pharmacophoric interactions, and this diagram which is shown all the interactions is known as pharmacophore. Okay. Now, next problem number 10 says cocaine was isolated as an active principle from coca leaves. Studies into SAR shows that the important functional groups for binding those are shown in blue, which is here. Okay, what is the binding interaction likely for the aromatic ring? This aromatic ring. Number A, hydrogen bonding as hydrogen bond donor. Number B, hydrogen bonding as hydrogen bond acceptor. Number C, van der Waals interactions. And number D, ionic bonding. Studies into the SAR related to the cocaine shows that the important functional groups are shown in blue in this diagram. What is the binding interaction are likely for the aromatic ring, which is here. So, this aromatic ring will show which kind of binding interaction. Hydrogen bonding as hydrogen bond donor, hydrogen bonding as hydrogen bond acceptor, van der Waals interaction or ionic bonding. Let me check the chat box. Okay, so the options are number C, number C, number C. So, all the options says it is number C. Now, let us see it. Okay, so the correct answer will be number C. Very good. Van der Waals interaction. So, this phenyl ring will participate in the van der Waals interaction. So, as you can see, there are various interaction can happen. This is the van der Waals interaction, means there are pi orbitals, okay, of this particular pi electron. So, this pi interaction is known as the van der Waals interaction, and it can also take part in induced dipole interaction if the ion is present, a positive ion is present here, because we have the pi negative charge of a pi cloud, and if the ions are present in between, so it can participate also in a induced dipole interaction. So, intermolecular interactions is known as pi stacking or pi interaction. It has 0 to 10 kilocalorie per mole energy. So, it is a weak electrostatic interaction between aromatic rings. There are two general types. One is face to face and another is edge to edge. Okay, so this is known as face to face when though pi or though benzene rings are like this. Okay, face to face, and there is face to edge when one ring is here and one ring is perpendicular to it. Okay, so this is called the edge to edge. So, face to face pi stacking interactions are responsible for the slippery feel of graphite. Similar pi stacking interactions helps to stabilize the DNA double helix. Okay. So, now moving toward the next problem. Problem number 11 says incorrect statement among the following is: number A, lesser the value of ED50, the drug will be more potent. Number B, larger the therapeutic index, safer is the drug. Number C, therapeutic index does not depend on LD50. Number D, lower the IC50 value, better is the drug molecule. So, identify the correct statement among the following. Try to write which of the following statement is correct. okay, or means which of the following is incorrect means what is the correct option. Then after your trial I will discuss what is ED50, what is LD50, what is therapeutic index and what is IC50. I will discuss all in detail, but before that all of you try to answer this question in the chat box. What is the incorrect statement among the following? Number A, lesser the value of ED50, the drug will be more potent. Number B, larger the therapeutic index, safer is the drug. Number C, therapeutic index does not depend on LD50. Number D, lower the IC50 value, better is the drug molecule. So whenever there is a confusion regarding this kind of true false question, try to rule out which options are incorrect. Okay, first try to rule out the incorrect options, then try to find out which of the for particular options is correct. Let me check the chat box. Please show question. So is the question was not visible? Was the question visible? Let me know in the chat box. Is the presentation is visible to you? Is the question number 11 is visible to you or not? Okay, others, Rabindranath. So I think, okay, I think it is a internet issue from your side. Please try to, okay, fine, I will discuss and please let me know, okay. So the correct answer will be number C, therapeutic index does not depend on LD50. Now let us discuss. So I think the presentation is visible, okay. Anyways, you can rejoin if you think there is an internet issue, you can rejoin if you cannot see the PPT. Okay, so the incorrect statement among the following is number C, therapeutic index does not depend on LD50. This statement is incorrect. Now let us discuss. What is therapeutic index? So the therapeutic index is the ratio between the dosage of a drug that causes toxic or lethal effect and the dosage that causes the therapeutic effect. Like therapeutic index in human, it is the ratio of TD50 and ED50, okay, and in animals it is the ratio between LD50 and ED50. Now what is ED50? It is the median effective dose, is the dose that produces the therapeutic effect in 50% of population. It is known as ED50. And TD50 or LD50, TD50 is the median toxic and LD50 is the lethal dose. Okay. So ED50 is the median effective dose, is the dose that produces the therapeutic effect of 50% of population, TD50 is the median toxic, the dose that is toxic to 50% population, and LD50 is the lethal dose that is toxic for the 50% of population. So, for human we take the TD50 value, means the median toxic ratio with the median effective dose ratio, and in animals we take LD50 and ED50 for the ratio. And what is the IC50? Basically IC50 is the concentration of a particular drug which is necessary to inhibit a particular drug target's action by 50%. So, if the lower is the IC50 value, means the lower is the concentration of the drug which is requiring for inhibiting a particular drug target, so better is the drug molecule. Okay. So, option number A is correct. Lesser the value of ED50, drug will be more potent. Okay, means we are requiring lesser dose to affect a population therapeutic effect in 50% of population. Number B, larger therapeutic index, safer is the drug, which is also correct. Therapeutic index does not depend on LD50, it is incorrect, because we see therapeutic index in animal depend on the LD50 or lethal dose 50, and lower the IC50 value, better is the drug molecule, it is also correct. So, it was the problem number 11. So, now problem number 12 says, which of the following areas is not a part of preclinical trials during the drug discovery? Your options are drug metabolism, number B pharmacology, number C toxicology, and number D structure and activity relationship or SAR. So, which of the following areas is not a part of preclinical trials during drug discovery? Drug metabolism, pharmacology, toxicology, or structure and activity relationship. Which of the following areas is not a part of preclinical trials during drug discovery? Try to answer this question in the chat box. Which of the following options A, B, C or D you think is correct? Okay, I will wait for some time for others to answer. So, which of the following is not a part of preclinical trials during the drug discovery? Number A, okay. Number A, drug metabolism, number B pharmacology, number C toxicology, and number D structural activity relationship or SAR. Let me check the chat box. Okay, number D is given by most of the students. Okay, let me show you the answer, then let us discuss. So, the correct answer will be, yes, it is number D, structural activity relationship or SAR is not a part of the preclinical studies. So, what is preclinical trial or testing? Preclinical trial is a laboratory test for a new drug or a new medical device, usually done on animal subjects to see if that hope for treatment really works or if it is safe to test on humans. Many preclinical tests include pharmacokinetics, the study of how drugs move through the living organisms. 4 processes are examined in pharmacokinetic studies. There are absorption, distribution, metabolism and excretion. Other tests that include chemistry test on purity, stability, shelf life of the product, as well as development studies like on dosing, packaging, administration, like through tablets or injection, etc. So, the drug metabolism, the pharmacology, toxicology, these are all part of the preclinical studies, but not the structural activity relationship. This is the part of the basic research or the designing part. Okay, the SAR is done on the designing part or the basic research part, but during the preclinical study only laboratory test happens on the animal subjects. Okay, to see to check if the compound or the drug or the medical device is safe for to use on the humans. So, now I will move toward the next problem. Problem number 13 says, which of the following statements regarding neurotransmitters is not true? Option A, they are secreted by neuronal cell. Number B, they are chemical messengers. Number C, they basically traverse through blood to act on a distant target cells. Number D, they carry out a cascade of reaction. So, which of the following statements is not true regarding the neurotransmitters? Read all the options carefully and let me know in the chat box which of the following you think is the correct answer. Means which of the following statement is not true regarding neurotransmitters. Number A, they are secreted by neuronal cells. Number B, they are chemical messengers. Number C, they basically traverse through blood to act on a distant target cell. And number D, they carry out a cascade of reactions. Everybody try to answer the question in the chat box. Let me check the chat box now. Okay, so I have few answers. Number C, number D, yes D, but through neurotransmitter reactions occur. Okay, let us discuss and then let me show you. Okay, so what is the correct answer? So incorrect statement is number C. They basically traverse through blood to act on a distant target cell. This is not correct. Now let us discuss. So neurotransmitters are chemical substances released by electrical impulses into the synaptic cleft from the synaptic vesicles of presynaptic membrane, then diffuses to be postsynaptic membrane, binds to and activates the receptors present, leading to initiation of new electrical signals or inhibition of a postsynaptic neuron. There are various neurotransmitters like adrenaline, which is produced in a stressful situation, increases the heart rate and blood flow, leading to physical boost and heightened awareness. GABA or gamma aminobutyric acid, which calms the firing nerves of the central nervous system. High levels of improved focus, low levels cause anxiety, also contributes to the motor control and vision. We have noradrenaline, which is the concentration. It affects the attention and responding actions in the brain, contracts the blood vessel, increasing the blood flow. Acetylcholine is with learning. It involves the through and learning and memory, activates the muscle action in the body, also associated with attention and awakening. Dopamine is for pleasure, feeling of pleasure, also addiction, movement and motivation. People repeat behaviors that lead to dopamine release. Glutamate is for memory. More common neurotransmitter is glutamate. It is involved learning and memory, regulates the development, creation and nerve contacts. Serotonin is for mood, contributes for well-being and happiness, helps sleep cycle, digestive system regulation, affected by exercise and light exposure. And we have endorphins, which are related to euphoria, released during exercise, excitement and sex, producing well-being and euphoria, reducing the pain. So these are the various neurotransmitters. So we have neurotransmitters and then we have hormones in our body. So both of these are made to seem like very similar in various aspects, but these two are very much different. Okay. So hormones are released by endocrine glands, neurotransmitters are released by nerve cells. Hormones travel through the blood to act on a distant target cells. Neurotransmitter travel across the synapse to act on nearby cells. Okay. So number C option, which says they basically traverse through the blood to act on distant target cell, it is not true. For neurotransmitter, they actually travel across the synapse only to act on the nearby cells, not the distant cells. Okay. The effects of hormone can be slower to build but long lasting, and the effect of neurotransmitters can be fast to act but shorter lasting. Hormones are part of a endocrine system and neurotransmitter are part of a nervous system. So, these were the hormone versus neurotransmitter, which is very important because both of them seems to like very related, but these are very different. Okay. So, the which the statement is not true is number C. Now, I will move toward the next problem. Problem number 14 says low concentration of neurotransmitter dopamine has been implicated in Parkinson's disease and L-DOPA is the most widely used medicine to slow down the disease. Which of the following statement is incorrect? Your options are: number A, dopamine cannot cross blood-brain barrier; number B, L-DOPA acts as a prodrug; number C, L-DOPA can cross blood-brain barrier; and number D, L-DOPA first decarboxylated by dopamine carboxylase before entering the brain. So, which of the following statement is incorrect? Read the options carefully, then try to answer. Low concentration of neurotransmitter dopamine has been implicated in the Parkinson's disease and L-DOPA is the most widely used medicine to slow down the Parkinson's disease. Which of the following statement is incorrect? Number A, dopamine cannot cross blood-brain barrier; number B, L-DOPA acts as a prodrug; number C, L-DOPA can cross blood-brain barrier; and number D, L-DOPA first decarboxylated by dopamine decarboxylase before entering the brain. Sorry. Try to answer the question in the chat box. Let me check the chat box now. Number C, number D, number A. So, I have 3 different answers. Okay. Now, let us discuss. So, the correct answer is, means the statement which is incorrect is number D. L-DOPA first decarboxylated by dopamine carboxylase before entering the brain. It is not true. So, blood-brain barrier, which I have discussed earlier also, it is a membrane that controls the passage of substances from blood into the central nervous system. It is a physical barrier between the local blood vessels and most parts of the central nervous system itself and stops many substances from travelling across it. The BBB is permeable to alcohol and some heavy metals can cross the blood-brain barrier as well. So, what is the role of this thing? So, this is the levodopa or the L-DOPA. Okay. So, this is the blood-brain barrier. So, what happens in the periphery of blood-brain barrier? So, in the periphery of blood-brain barrier, the 2 enzymes, so first is the COMT. So, this enzyme converts levodopa to 3-methoxytyrosine, okay, and AADC, it converts it into the dopamine. Okay. So, this levodopa, it can cross the blood-brain barrier. As you can see, it can easily cross the blood-brain barrier. Okay. And when once it crosses the blood-brain barrier, it is converted to dopamine by the same enzyme AADC. Okay. And this dopamine then again converted to DOAPC by MyoB. So, as the if you read all the options now, first statement says that dopamine cannot cross blood-brain barrier. It is correct. Okay, dopamine it cannot cross the blood-brain barrier, but levodopa it can cross the blood-brain barrier. Okay, so levodopa it acts as a prodrug. Prodrug means it is not an active form of drug. Once it passes through the blood-brain barrier, it will convert it to the active form, which is the dopamine by the enzyme AADC. Okay, and then it will exert its exert its activity. So, the number D, option number D, which says levodopa is first decarboxylated before entering the brain, it is incorrect. Before entering the brain it is not decarboxylated. It can cross the blood brain barrier as it is, and then after that what it will do, it will convert itself into the dopamine. Okay. So, this was the problem number 14. So, now problem number 15 says beta blockers are used as dash agents. Now, fill in the blanks. Your options are number A, anti-diabetic, number B, anti-hypertensive, number C, antibacterial, and number D, antifungal. So, which of the following statement is correct regarding beta blockers, and what does it use for? Is it anti-diabetic? Is it anti-hypertensive? Is it antibacterial, or is it antifungal agent? Try to answer this in the chat box. Beta blockers are used as dash agents: anti-diabetic, anti-hypertensive, antibacterial, or antifungal. Let me check the chat box now. Okay, so I have few answers, number B, number B, number B. So, everyone thinks it is number B. Now, let us discuss. So, yeah, very good. The correct answer is number B, antihypertensive. So, beta blockers are used as antihypertensive agents. So, antihypertensive agents are basically used for high blood pressure, right? So, for high blood pressure we have antihypertensive drugs, and there are various kinds or various classes of this antihypertensive drugs. Now, let us discuss. So, first there is diuretics. Okay, what is the mechanism of action of diuretics? They act on kidneys to increase the excretion of sodium and water, decrease the blood volume, decrease the BP or blood pressure. So, what is the example of diuretics? There are thiazides like hydrochlorothiazide or chlorothianidone, high ceiling which are furosemide, K+spurin which are spironolactone, amiloride and triametrine. Okay. So, you do not need to memorize each and every name, but if you remember, just remember the name of the hypertensive agent. Okay. What is the hypertensive agent and mechanism of action of the particular reagent, like for diuretics what is the mechanism of action and one example of the particular class, it will be enough. So, what is the mechanism of action for diuretics? They act on the kidneys to increase the excretion of sodium and water, decreasing blood volume, decreasing BP. Angiotensin converting enzyme inhibitor or ACE inhibitor. So, ACE inhibitors are very important. Okay. ACE inhibitor are very important antihypertensive agents. Your examples are captopril, linezolid, enalapril. Okay. So, the mechanism of action inhibiting the synthesis of angiotensin 2, decrease the peripheral resistance and blood volume. Angiotensin 81 blockers. So, the examples are losartan, candesartan, valsartan. So, mechanism of action blocking the binding of angiotensin 2 to its receptors. And we have adrenergic receptors antagonists which are the beta blockers like propranolol, nebivolol, atenolol. The mechanism of action is competitive antagonists that block the receptor sites on the adrenergic beta receptor. So, these are the 4 classes of antihypertensive agents, their examples and also their mechanism of action. So, I will suggest remember this 4 class, remember the mechanism of action of each and every class and 1 or 2 examples of each drug. Okay? That will be fine for your syllabus. Moving toward the next problem. Problem number 16 says levodopa is used as prodrug for dopamine since it entered the brain more effectively than dopamine. Why? What is the reason due to which levodopa can cross the blood brain barrier more easily than the dopamine? Your options are number A, it is more polar and crosses the blood brain barrier more effectively. Number B, it is less polar and crosses the blood-brain barrier more effectively. Number C, it mimics an amino acid, takes advantage of carrier proteins in order to cross the blood-brain barrier. Number D, it is less susceptible to metabolism. So, levodopa, it is used as a product of dopamine since it enters the brain more easily than dopamine. I have discussed it in the last to last slide. So, what is the reason due to which the levodopa can cross the blood-brain barrier more easily? Read the options carefully and then try to answer it in the chat box. Which of the following you think is the correct option? Number A, it is more polar and crosses blood-brain barrier more effectively. Number B, it is less polar and crosses blood-brain barrier more effectively. Number C, it mimics an amino acid, takes advantage of carrier proteins in order to cross the blood-brain barrier. Number D, it is less susceptible to metabolism. Let me check the chat box now. Okay, so I have two answers which says it is number A. Okay, now let us discuss it. So, the correct answer is number C. It mimics an amino acid, takes advantage of carrier proteins in order to cross the blood-brain barrier. So, as you can see, this L-dopa, it resembles more like a tyrosine. Okay, if you see the structure of levodopa, the structure is almost similar as the tyrosine, only the difference is like there is one extra hydroxyl group here. Okay, so since it mimics the tyrosine, okay, this levodopa, and it can easily cross the blood-brain barrier. Okay, because it mimics the amino acid, and amino acids are can be taken up by this blood-brain barrier. So, how it happens? Tyrosine, it gives the levodopa and it converts to dopamine by DCC, dopamine decarboxylase. Okay, and we have this blood-brain barrier here, and then there is the presynaptic terminal from the substantial nigra. Then this tyrosine, it converts to the levodopa, and this gives rise to the dopamine. Okay, and this is the dopamine receptors here. So, this is the whole mechanism of action. So, remember, the levodopa mimics the amino acids and takes advantage of the carrier proteins in order to cross the blood-brain barrier. So, now I will move toward the next problem. Problem number 17 says, the term used to signify the biological effect of a preparation without the active drug that appears to induce beneficial effects similar to that of the active drug preparation is number A, placebo, number B, agonist, number C, me too drug, and number D, peptidomimetic. Read the question carefully, then try to answer this in the chat box. The term which is used to signify the biological effect of a preparation without any active drug that appears to induce a beneficial effect similar to that of the active drug preparation. So, we have a preparation which do not have an active drug, but it induces some beneficial effect similar to that of the active drug preparation. It is known as a placebo, a agonist, a me too drug, or a peptidomimetic. Try to answer this question in the chat box. Let me check the chat box. Okay, is my voice not audible? There is a little bit noise here, so please let me know if my voice is clear or not. Hello, is my voice audible or not? Okay. So yeah, there is some celebration due to the Ganesh Chaturthi. Okay, so that's why the voice is getting it is little bit hampered. So bear with me. Okay, now let us discuss the problem number 17. So the correct answer is number A, placebo. So placebo is the term which is used to signify the biological effect of a preparation without any active drug that appears to induce the beneficial effect similar to that of the active drug preparation. Okay, so what is placebo and what is the other terms? Let us discuss. So definition: a placebo is defined as a substance that has no known specific pharmacological activity against the condition being treated. Placebos can be therapeutically beneficial to some patients when they give rise to the placebo effect. Okay. So placebo effect, what is the placebo effect? A fake treatment can make someone better simply because they expect it to help. This will make you feel better. If this could be real or fake, I will feel better already, as long as they believe it will help. So placebo is basically, it is this component or this particular preparation do not have any active drug, but it is given to a patient to say, let us say like it is a particular treatment which will help you, and the patients believe that is the particular pharmacological effect which is shown by in the patient, which is known as placebo effect. So what is agonist? Agonists are drugs that occupy the receptors and activate them. Antagonists are drugs they occupy the receptor, do not activate them. Antagonists block the receptor and activation by the agonist. So that is agonist and antagonist, which are several different parts. So what is me too drug? A drug that is structurally very similar to already known drugs with only minor differences. The term me too carries a negative connotation. However, me too products may create competition and drive prices down. Me too drugs can be broadly defined as chemically related to prototypes or other chemical compounds which have an identical mechanism of action. And peptidomimetics incorporating conformational constraint locally or globally. So what is a peptidomimetic? These are molecules with significantly reduced peptide character that mimic the bioactive conformation of peptides and thus retain the ability to interact with the biological target and cause some biological effect. Small protein-like chain designed to mimic to peptide arise either from modification of existing peptide by designing similar system that mimic peptides, for example peptidoids or beta peptides. So these are the peptidomimetics. So these are the 4 terms which are mentioned here, and we have few more questions left. Please bear with me. I think my voice may not be clear for some time due to the noise, but I will try my best, okay, so that you can hear my voice. So problem number 18 says, which of the following is false regarding the renin-angiotensin system? Your options are: number A, aldosterone is a steroidal hormone; number B, vasoconstriction is caused by the action of angiotensin 1; number C, aldosterone enhances the retention of sodium and water in the blood; number D, bradykinin is destroyed by ACE or angiotensin converting enzyme. So which of the following statement is false regarding the renin-angiotensin system? Read the options carefully and let me know in the chat box which of the following you think is the correct answer. Which of the following statement is false regarding renin-angiotensin system? Number A, aldosterone is a steroidal hormone; Number B, vasoconstriction is caused by the action of angiotensin 1. Number C, aldosterone enhances the retention of sodium and water in the blood. Number D, bradykinin is destroyed by ACE or angiotensin converting enzyme. Let me check the chat box now. Okay, I have one answer which says it is number C. Okay, let us check and then let us discuss. So, the correct answer is number B. Vasoconstriction is caused by the action of angiotensin 1. It is false. Now, let us discuss. So, what is renin-angiotensin pathway and what is aldosterone? So, aldosterone is basically a steroid hormone, right? It is so from adipose tissue we have this aldosterone here, and it is related to the sodium retention and high blood pressure, okay? And due to high blood pressure there are oxidative stress, damage in DNA and vascular remodeling, okay, like arterial stiffening and arteriosis, renal fibrosis, okay, cerebrovascular disease, stroke. So, these are all related with the high blood pressure. So, what is the renin-angiotensin aldosterone pathway? So, this is basically related to the increase or decreasing of the blood pressure. So, first there is dehydration or sodium deficiency or hemorrhage, which is give rise to decrease in the blood volume. Then the blood volume decrease give rise to the decrease in the blood pressure. Okay. So, whenever there is a decrease in blood pressure, what happens? The juxtaglomerular cells of the kidneys are activated and they increase the renin secretion. Okay. And then we have this angiotensin from the liver, okay, and this increased renin secretion give rise to the increased angiotensin 1. Now, this angiotensin 1 is converted to the angiotensin 2 by this angiotensin converting enzyme which is present in the lungs. Okay. For in the lungs, angiotensin 1 is given rise to the angiotensin. Angiotensin 1 is goes to angiotensin 2, and then angiotensin 2 it related to the vasoconstriction of the arterioles, okay, and it is also goes to the adrenal cortex, which increase the amount of aldosterone. Increased amount of aldosterone in kidneys increase sodium and water reabsorption, which increase the blood volume and increase the blood pressure till the blood pressure comes to normal. Okay. So, this is the all ranging angiotensin aldosterone pathway which goes on and on whenever there is a lowering of blood pressure or there is a increase of blood pressure. Okay. So, vasoconstriction is caused by the action of angiotensin 1. It is incorrect because vasoconstriction is actually related to the angiotensin 2. That is why whenever there is a hypertension, inhibiting the angiotensin converting enzyme is a very good treatment option because it actually inhibits the conversion of angiotensin 1 to angiotensin 2. Okay. So, this was the renin-angiotensin system. So, quickly I will finish today. So, this is the problem number 19, which says which of the following statements is true regarding the cholinergic receptors. Your options are: number A, the inner part of cell membrane is hydrophilic in nature. Number B, the cholinergic receptors are of 3 types. Number C, the nicotinic receptors activate the ligand gated ion channel opening. And number D, acetylcholine esterase enhances the effect of the acetylcholine. So, which of the following statements regarding cholinergic receptor is true? Let me know in the chat box. Let me know in the chat box which of the following statement is true regarding cholinergic receptor. Let me check the chat box. Okay, let us discuss that, okay, because there is a very sound problem here. So, I will quickly finish off today. So, the correct statement will be number D, acetylcholine esterase enhances the effect of acetylcholine. So, this is acetylcholine receptors, okay, which is the muscarinic acetylcholine receptors and nicotinic acetylcholine receptors. Nicotinic acetylcholine receptors are of two types, one is neuronal type nicotinic receptor and one is muscular type nicotinic receptor. So, acetylcholine receptor stimulation of all autonomic ganglia, stimulation of voluntary muscle, secretion of adrenaline from adrenaline medulla. Acetylcholine nicotinic receptor fall into three main classes, the muscle, ganglionic and CNS. These are typically the ligand gated ion channels. Muscle receptors are confined to the skeletal neuromuscular junction. Ganglionic receptors are responsible for transamination and sympathetic and parasympathetic ganglia. CNS type receptors are widespread in the brain. So, option number D is correct. Acetylcholine esterase they enhances the effect of acetylcholine. Okay. So, the last question of today's session is problem number 20, which says the correct statements amongst the following regarding the antihypertensive drug enalapril is: number A, it is an irreversible inhibitor of angiotensin converting enzyme. Number B, it is a prodrug. Number C, it has a good bioavailability. And number D, it is a transition state analog inhibitor of ACE enzyme or angiotensin converting enzyme. Try to answer this question in the chat box. Which of the following you think is the true regarding the antihypertensive drug enalapril? Number A, it is a irreversible inhibitor of angiotensin converting enzyme. Number B, it is a prodrug. Number C, it has got poor bioavailability. And number D, it is a transition state analog inhibitor of angiotensin converting enzyme. Let me check the chat box, then let us discuss. Okay, I will quickly discuss it, okay, because so the correct answer will be number B, it is a prodrug. It is the correct statement regarding the enalapril, okay, which is an antihypertensive agent. So, what is enalapril? It is an angiotensin converting enzyme or ACE inhibitor. It is a prodrug which when hydrolyzed by esterases to is active from enalaprillate. Mechanism of action: enalaprillate competes with angiotensin 1 for binding at the angiotensin converting enzyme, blocking the conversion of angiotensin 1 and angiotensin 2. As angiotensin 2 is a vasoconstrictor, a negative feedback mediator for renin activity, lower the concentration results in decreased blood pressure. Enalaprillate may also act as a kinase 2 which degrades the vasoconstrictor bradykinin. So, this is the enalapril, which is a prodrug which can cross the membrane. Enalaprillate, it is a active hypertensive agent. Okay. So, enalapril is a ester, as you can see it is a ester form and it can easily cross the barrier. Enalaprillate is a acid form which is the active form. So, enalapril is a prodrug and enalaprillate is the active anti-hypertensive agent. So, this was the last question of today's session. So, today we discussed about the drug development processes, then we discussed about the neurotransmitters, the L-levodopa and then acetylcholines, and then we discussed about anti-hypertensive agents. So, it was the week 8 live session for this NPTEL course of Organic Chemistry and Biology and Drug Development, where we discussed the week 9 course content. So, I will share my email ID with you. If you have any doubt, any query regarding today's session, feel free to send me an email to this email ID which I am sharing. Okay. Now, I will try to answer your query through the email. If you have any doubt, any query regarding today's session, it was the week 8 live session for this NPTEL course. Next Tuesday we will have this kind of sessions till 12 weeks you are completing. So next Tuesday we will have this kind of session, and then we will discuss the week 10 course content. So from now on, from this week, we have moved toward the drug development processes and the drug agents and all of these things we have discussed. Okay, and we will more discuss in upcoming weeks various drug agents like anti-cancer agents, antibiotic agents in detail in upcoming weeks. So do not miss the classes in upcoming week. Okay, it will be beneficial for you for your course. And thank you all for joining and keeping up with me till now. And I will share this PPT and also this class material with you soon. And if you have any doubt, any query, feel free to contact me. Please join in the next week's class also. I will close the session for today, and thank you all. If you have any query, feel free to reach. So, okay, I will close the session for today. Thank you all.